Welcome to the BEL Webinar Series for 2023. We are excited to share new research and ideas in the neuroscientific community. Today's webinar is Electrical Perturbation of Human Cortex Generates a Reliable, Individualized Readout of Cortical Excitability. What's the potential, and is this just a phase? Dr. Allison C. Waters, PhD, and Assistant Professor of Psychiatry and Neuroscience at the Nash Family Center for Advanced Circuit Therapeutics, Icahn School of Medicine at Mount Sinai, is our presenter. Please write your questions into the chat feature and we will answer them during the Q&A period. And now I will turn it over to Dr. Waters.
Thanks so much, Corinne. I see some familiar names on the line today. And thank you for showing up. I'm so excited about this line of research and would love to be sharing it and discussing it with the electrophysiology community here. So let me put my slides up. Some of you may know that I did my doctoral training with Dr. Tucker here at the U of O, and also at the Millrace Research Park. So you can imagine it's pretty thrilling for me to get to double back and tell you a little bit about what I've been working on. I'll start with disclosures. I don't have any disclosures that are relevant to this talk, but shout out to the NIH, I guess, and the Brain and Behavior Research Foundation and the Hope for Depression Foundation that are supporting this work, and also close collaboration with all of these PIs at the Icahn School of Medicine. Deep brain stimulation is very interdisciplinary, so we've all got to work together and leverage expertise from a variety of domains. So that's what I'm going to talk about today, deep brain stimulation. I'm going to start by telling you a little bit about how we target in the brain for DBS with DBS, but really, that's just setting me up to be able to talk about a bigger problem that we have. We call the target engagement problem. And one way we're attacking this problem with stimulation evoked potentials, which is just a strategy that we're developing to optimize treatment with deep brain stimulation. For those who don't know, deep brain stimulation, or DBS, it's an invasive surgical strategy. The leads are placed in some target, some zone of the circuit, and then a wire is fed all the way down into the chest where there's a pacemaker that controls the current. We use deep brain stimulation as a psychiatric intervention for Tourette, for OCD, for depression. But the mainstay is movement disorders. And at our center, we do all those diagnostic indications and build a whole research kind of community around that clinical practice in an effort to borrow insight from one indication to help kind of leverage or optimize or innovate the treatment for other indications, emerging indications. So yeah, today I'm going to be talking a little bit about perturbation mapping, and I was thinking for this group, a goal would be that if you want to start using this technique in your research, I can give you a little intro, and then you can DM me, and I'll share some tips. But also, I suspect that this is an emerging brain mapping technique, and that it's going to become a mainstay, not only in neuromodulation, but in basic science as well, a way of understanding the brain in terms of cortical responsivity to stimulation. And as it emerges and finds new applications, I think the more we can talk about it in a forum like this, then the more we can sort of celebrate it together and marvel at the emergence of this. Certainly, the future is electrophysiology, but you wouldn't be on the line if you didn't know that already. Okay, so Let's talk a little bit about how deep brain stimulation for depression is targeted, why we put the stimulator or why we put the electrodes in an area of the brain called the subcallosal cingulate. It's a fascinating discovery narrative. It's a great story of science, how it moved from one idea to another idea as new information came in. It actually began in the functional domain, really in the early days of fMRI and PET, with the observation that if you induced profound states of sadness or studied the brains of individuals with severe depression, this one region of the brain that I'm showing here, the subcallosal cingulate, was hyperactive. Moreover, if you treated that severe depression or reduced that profound sadness, you would reverse that effect. So that was Helen Mayberg's original hypothesis. Oh, now that I think back, Dawn, I could have shown some of your work with Liotti, who's probably up at UBC now, right?
Yeah.
Oh, yes. So you were there too, pointing to this region.
Yeah.
Yeah, so Dr. Mayberg borrowed then from movement disorders where they were already starting to interrupt pathological signaling in the basal ganglia and put a deep brain stimulator there. And miraculously, people with very profound depression started showing some changes. And then after that, there was a bunch of hype. So everybody found reasons why this particular area was somehow related to depression and depression pathology. So the thing is, they put the stimulator in, these are in small samples, and maybe half the sample would get better. And so then the next phase of this science was to try to optimize, to figure out why some people were getting better and some people weren't. And the neurosurgeons needed a guide. They needed to know where in this, relatively large target zone, the subcallosal cingulate, to put the active contact, or where you'd be injecting the electricity. And it turned out that mere stereotaxic coordinates, so that's that picture that I have with the dots, all the different targets for different patients, mere stereotaxic coordinates weren't sufficient to tell us who was going to get better and who was not going to get better with the treatment, or to tell the neurosurgeon exactly where to put the lead. This is a problem, right? Because you're asking for an individual to undergo invasive a brain surgery, and you really want to know you're getting it in the right place. So that was the focus probably for about 7 or 8 years, just figuring out the right place and enter this insight or connectomic research. But something that I think is interesting about the turn to white matter as our mapping strategy is that it coincided simply with the emergence of the technology, right? So we had a hammer and we found a nail, but it's been a very productive nail. So let me tell you what I'm looking at here. It turned out that when you mapped the DTI, the white matter connectivity profile, from the active contact, the therapeutic contact, in people who got better, well, all those people had something in common. What they had in common was that the active or therapeutic contact was sitting right at the confluence point of these 3 white matter fiber bundles that pass through the subgenual region. The forceps minor, the uncinate fasciculates, the cingulum bundle. So then the thinking about targeting shifted entirely from functional to structural. It is the confluence point that we're targeting. What made this theory convincing was that they then went to the folks who had not responded to treatment. They ran the DTI maps and they moved the therapeutic contact to that confluence point, and within 6 months, those individuals got better. So that was pretty convincing. Definitely ushered in another 5 years of sort of obsessive work in the connectomic domain. But really where the most convincing evidence is in the clinical outcomes. So here I'm showing 3 consecutive cohorts, blue, then red, then green. In this group that was using connectomic targeting and getting better and better at leveraging the connectomic maps, making better DTI models, having better communication with the neurosurgeon. And you could see that individuals got better faster. And I'm only showing the first 4 weeks following the surgical implantation, but they stayed better longer as well. So a pretty radical change in these folks, given how sick they are. But at the very same time that this group was seeing some success with enhanced targeting of their deep brain stimulation, a randomized multi-site clinical trial was halted for futility. So that's a little bit different from coming up with a null result or a failed trial. Halted for futility means that at an early time point, the active treatment group didn't differ enough from the placebo group to warrant a continued study, given how much it costs to do this kind of work and the risk. There was a pretty high bar there. But if you see my targeting treatment outcomes chart there, you can see why, when we began discussing why this trial was halted for futility, everybody was talking about target engagement. It must have been that at various sites, they were doing it in all sorts of different ways. Some were using connectomic methods and some were not. But needless to say, that was a real blow to the community and to the potential for this treatment to be disseminated more broadly. And, it's a life-saving treatment for people who really have no other options. So this is where we find ourselves. We know this works. These are the long-term outcomes out to 8 years following implantation. 50% remission, 70% responding, and that is an extraordinary outcome for folks who have a very high mortality rate, who have been chronically ill their entire lives, failed ECT, failed TMS, failed all of the SSRIs and psychopharmacological intervention, CBT. So how to disseminate? How to develop target engagement biomarkers that we can disseminate to other sites and teach them, what we're doing, so that we can run another trial, so that we can, make this treatment a bit more broadly available. The big challenge for psychiatric DBS is that we don't have the same acute behavioral response that you see in movement disorders. Acute behavioral response to stimulation that the neurologists use to target and tune the stimulator right there in the clinic. So if you haven't seen this before, I'm just going to take a moment to show you what I mean. I just found a YouTube clip. I usually use one from, let's see if I can. Can you guys see my YouTube? Yeah, okay, good. Yeah, I just picked this off of YouTube. You can find them anywhere. But let's just watch and see.
The initial programming session. You got both sides of the brain put in, bilateral stimulators, and we're here for the initial programming session. This is what it looks like without the medicine and the stimulator. Now we're just going to turn it on. I can get my fat fingers to hit the button. There we go. See how that feels. How you feel?
Yeah, a lot better.
Yeah. Excellent. Try tapping both your fingers for me. Excellent. All right. Feels pretty good?
Oh, yeah.
Okay, good.
Right. In movement disorders, profound acute response. And what the YouTube clip is not showing is that the neurologist is going to be hunting around for that treatment response. They'll be changing exactly which contact they're using or combination of contacts, their source sync configuration, their amplitude, their pulse width. We're playing around with all these different parameters, stimulation parameters, to try to find that sweet spot, which is now more broadly defined, not just as a spot, a location, but a whole set of parameters. We don't have this in psychiatric disorders, partly because the symptom change we're looking for probably lags behind the brain change, right? Now, we have reported some acute change in the operating room. So I have a couple examples of those up here, the Choi and Rivapose, where it's fascinating. The patient, when we're on the spot that will ultimately be used for successful therapy, they report a sort of interoceptive enhancement. They might describe feeling warmer or becoming more aware of their body or in their body, but they'll also potentially engage in some social interaction, like look over to their, well, they won't look because they're in a head frame, but they might acknowledge somebody who's in the room who's been there the entire time, but only now they recognize them. So we aren't able to replicate this in the laboratory or in the clinic, or we don't see it with everybody, but it is a clue. So that gives us 3 different strategies that we can use to derive target engagement biomarkers, working on these in the lab. The first approach is, this decoding, finding correlates of treatment response, correlates of psychiatric constructs, just like we do in movement disorders. A second approach is to understand these interoceptive effects and chase them, see if we can induce them. And a third approach, what I'm going to talk about today, is perturbation mapping. The idea here is that we can speak directly to the cortex, right? Like, what if we had a way we could circumvent this need for the behavioral response to guide us, to tell us we're in the right place? What if we could communicate directly to the cortex? So we and others are proposing this approach. We call it a ping or a perturbation map. A probe of the cortex, where you hit it with a little bit of current at a given parameter set, and then you watch what the cortex does in response. And it's miraculous. It really is a very coherent spatial temporal pattern that you can see on the head surface that is telling us how the individual's cortex is responding to STEM at that given parameter set. You can use invasive or non-invasive methods to create a STEM evoked potential, deep brain stimulation or TMS. You can use invasive or non-invasive measures of the electrocortical response, and each have their advantages and disadvantages. So we're all working together to try to understand the biophysics of this effect. For me, it's most important to use a non-invasive recording method so that I can track patients over time in the clinic, so that I can bring them into the clinic situation and use the perturbation map to guide target engagement. And also we want to be looking at how target engagement might change over time in treatment with DBS as the brain adapts and changes to the stimulator itself. It's also an area that we're going to be collaborating with BEL to innovate a little bit more in the source modeling space, which is the other advantage of working on the head surface. It's really the only way that you can get the whole cortex view and begin to understand where in the cortical architecture these signals are emanating. So for the next four or seven slides, I'm going to take you through some of the work that we've been doing to understand this signal really so that we can later exploit it for our use case, but there's a lot of engineering work that still has to be done to understand, to be able to interpret this signal. So when I stimulate the SCC region specifically, I get a signal that looks like this. It rolls through really 4, maybe 5 components between 15 and 150 milliseconds. I'm showing them there for those of you who are unfamiliar. I'm sure this is a technique that a lot of you are using on the line. So we're looking down at the head surface with the nose pointing to the top of the screen there with those circles, with the imagining the head as a sphere. So you can see the voltage patterns there are wildly coherent. Moreover, they're so reliable. They're almost like a brain print. If I scramble a group of patients' perturbation maps from different sessions, I can easily classify them back into the patient-specific groupings. And most important for my purposes, the stimulation evoked potential is very sensitive to changes in stimulation parameters, like which contact on the lead I'm stimulating from, the amplitude, the pulse width, even the waveform. So talking about this specificity, another stunning design characteristic of this STIM-EP is how it captures individual differences. I like to put this picture up here just to think about the metaphor of faces, how we all have 2 eyes, a nose, and a mouth, but of course, the face itself contains so much individual difference that characterizes who we are, right? Same for these perturbation maps. So here they're going to run through you'll be able to pull out the features that I showed you on the screen before. These, I'm going to walk you through it again. Okay, so I'm looking at 6 different individuals here. They're all getting perturbations maps from their subcallosal cingulates. And what your eye might be drawn to are the first 4 features, sweeps down the midline, a posterior positivity, and then becomes more diffuse. But what you see also are these individual differences, how curious. And our work is really to discover what is driving that variance so that we can leverage it as a means to move through the cortex guided entirely by the cortical response to stimulation at any given parameter set. Now, what I'm showing down there in the inset is stimulation to a different region of the brain entirely, the subthalamic nucleus. So it gives you a clue that where you're landing in the brain is going to give you a very specific perturbation map. Let me show you it from a different point of view. So here are 3 perturbation maps from 3 different regions of the brain, all targets of DBS for different diagnostic indications, SCC for depression, anterior limb of the internal capsule for OCD, subthalamic nucleus for Parkinson's disease. So we can begin to imagine when we look at these different perturbation maps, how an effective connectivity connectome might converge if we were to work together as a community and sort of create a database of different spatial patterns emerging from stimulation at different locations in the brain, so that we can decode, I would say, the biophysical properties that give rise to this pattern and extract from it meaningful indices that we can apply to a variety of problems. So back to our target engagement problem. An obvious first start for us in terms of decoding the perturbation was white matter. We wanted to know if these patterns on the head surface could be a readout of or a correlate of white matter connectivity. It makes sense, right? Because you're stimulating white matter, at least according to the current working definition. And so you would imagine that that would be a way that the signal would propagate around through the brain, right? But we're actually way out 25 milliseconds, 150 milliseconds following stimulation. So this is well beyond a monosynaptic volley. So actually the jury's still out. The concept is there, but there's a lot more. There's a lot more information contained in a perturbation map than just that kind of one-to-one white matter mapping. Anyway, our early findings or our early observations were promising. Here I'm showing 3 patients in a row that had this one bilateral feature when I stimulated on a contact or an adjacent contact, but then one patient in particular, that bilateral feature showed up as just on one side. So when we looked in the white matter, and of course, we also found in their white matter map that the connectivity across the corpus callosum via forceps minor was a little less. We weren't getting into the frontal lobe on the other side. So it was a good clue. And so as a next step, we extracted the magnitude of that one feature, the P40 feature, and we plotted it relative to the percentage of white matter that was being perturbed in that volume of tissue that was activated by that given parameter set. And we found that the magnitude of that one feature, the P40, was actually a pretty good predictor of how much forceps minor we thought was being perturbed by stimulation at that parameter set, but a rather poor predictor of the cingulum bundle, which moves in a different direction and might have a different pattern on the head surface. It's a bit of a tortured inquiry. We really have to work hard to see the relationship between the perturbation map and white matter, but it is there. So let me tell you about this study. I think this is really interesting. It's a collaboration with my colleagues at Duke. And we'll also be working with BEL on this project to run the neural source modeling. So the way we designed this study was to step back a little bit from the treatment mechanism itself and leverage advances in white matter modeling called field cable modeling, where we can specify, using these white matter models, the set of DBS parameters that might activate or perturb one fiber bundle at a time. So in the top 2 rows, we're using stimulation settings that we think are going to affect the cingulum bundle alone. And in the bottom 2 rows, stimulation settings that we think are going to affect the forceps minor, moving into the frontal lobe there. You can see for yourself that the perturbation map is strikingly different. It's almost like as if there is a signature specific to cingulum stimulation and another signature specific to forceps minor stimulation. So we'll have to move into the source model to understand whether our hypothesis bears out in cortical architecture. And Andreas will be taking the lead on that. He's got a forthcoming manuscript showing these particular results. We're looking forward to sharing that. Meanwhile, we're in electrophysiology space, and we're recognizing that these perturbation maps actually offer an even richer view of what we would call circuit dynamics. What I've been showing you so far uses really conventional feature extraction the kind of feature extraction method that you would see in a cognitive or affective evoked response, right? Where we average across time, or we average across multiple trials to reduce the noise. But when we do that, we're not fully exploiting what may be the most important part of a circuit dynamic and what we really need to be listening for in constructing these target engagement biomarkers. It's hard to do in this context because you need a really clear signal. And in the laboratory, the patients can't tolerate a lot of recording time. But we're starting to make some progress here, enough to show that the largest component of that perturbation map is likely caused by realignment of the theta phase in frontal channels, at least. And this theta phase alignment is somewhat predictive of that white matter fiber bundle again that's traversing into the frontal lobe. So let's step back a little bit, maybe for the last few slides of my talk, and let me tell you a bit about our progress thus far in devising a target engagement biomarker using this strategy. I'm in an interesting position working at Mount Sinai at the Center for Advanced Circuit Therapeutics. Mount Sinai's motto is "We find a way." So I actually had this slide as a little tip of the hat to my colleagues here who have outcomes that are so good in the 80 to 90% range. Really, they're getting people better there. So they're doing something right. And whether it's a connectomic targeting that is the thing they're doing right or not, doesn't really matter. It gives me an incredible experimental opportunity to just work with a population of people who, for whatever reason, the treatment is working for them. So then it lets me ask a question. What is so special about the parameter set and the location where the people do get better? What is so special about the therapeutic contact or the on-target contact compared to other contacts? Here at the head surface, a little bit disappointing, nothing particularly special. What I see is that as I move from the ventralmost contact in the target region to the dorsalmost contact, the amplitude increases. But this wouldn't tell me, if I was trying to program as a neurologist, that I'm on the sweet spot. It turns out that I have to move into neural sources to see that effect. So it turns out that you have to deconstruct the cortical signal into source space in order to see that differentiation that is actionable, at least in this particular clinical application. So here I have pulled a source ROI from the area that was most implicated by all the white matter investigation. And if I plot it, the time course of the evoked response, comparing adjacent contacts to the contact that got everybody better, that's in red, the therapeutic contact, I see in this cohort that it seems to have a faster latency. So it arrives when you're on the right spot, it arrives in that frontal ROI a little faster than if I targeted or if I stimulated in other parts of the target region. Then I ran this experiment again on a completely different cohort at a different hospital. They even had a different device type, and I saw it again. So it's pretty convincing that what we have here is a method for confirming that I've picked the right contact among many contacts to choose from, where where all I would really need is a few electrodes on the forehead to measure that temporal difference. To the extent to which this relates to theta phase alignment, we have yet to see, but that would be a good hypothesis to test. So I think I might stop there. I prepared a few slides for to talk about OCD, but I think maybe I'm going to stop because I'm curious to know what kinds of questions are coming up for the audience here, and I can pull those OCD slides up if that is something you would like to talk about. Okay. And some acknowledgements to my team at Sinai.
Thank you so much for that lecture. We do have a question in the chat. Is the early response in slide 20 for an on-target explainable as a phase shift of the frontal theta rhythm? Okay, we have a couple questions in the chat here. So this is from Mike Posner. What are the optimal stimulation parameters that you have found so far?
Can you repeat the question?
Yeah, so we have a question from Mike Posner. What are the optimal stimulation parameters that you have found so far?
Okay, so once the location has been determined, we found a pulse width of 90 microseconds and an amplitude usually around 4.5 volts. But sometimes for some people, we have to move up between 4.5 and 6 volts. Did you have anything in mind in particular?
Do you use a single pulse or a rhythmic pulse?
130 hertz.
Oh, okay.
So your point there, or your question there, points to a way in which what I've been talking about, perturbation mapping, is very much a technology. So it's not a readout of the treatment mechanism per se, but more a readout of cortical responsivity to stimulation that we're then exploiting in the context of this treatment.
Great. And then we had another question. Is the early response in slide 20 for on target explainable as a phase shift of the frontal theta rhythm?
That is an excellent question. That would be a great hypothesis to test, because we've seen that phase alignment as the main contributor to the average ERP. So, yeah. And that would be really, that would be a thrilling finding, but also fitting. Theta has been implicated in, midline theta has been implicated in depression and depression recovery in a variety of cases. Some struggle to replicate that stuff, but it's there, right?
Great. And another question is, are there any short-term and or long-term side effects arising from the treatment?
Side effects. So this is an invasive treatment, so there's a significant recovery period. In movement disorders, we do work around a lot of side effects, but in psychiatric disorders, in depression, this has not been a problem. In OCD, though, this is an interesting part of the current debate around targeting. Some groups actually target the nucleus accumbens or some ventral areas of the target that is agreed upon at this time, and it tends to actually make people a little bit manic. So some groups see that response as a side effect, and we try to avoid it. Other groups actually think we can leverage a little bit of hypomania to help people out of their intractable OCD. So that's an interesting case. That's an interesting case where there's debate over whether we should even define that effect as a side effect.
Great. Here's another question. Do you think more effective target engagement mapping will become a standard practice for DBS treatment and depression?
I do. I think this is the point that we're at in the discovery narrative in innovating in this area. I think that we and others are working on this problem, and it's the key gap between where we're at right now with a halted trial and the likelihood that the FDA will approve this as a treatment.
Great. Well, unless there are any other questions in the chat, I just want to thank you so much for delivering this webinar. And sorry for all of the trouble with the accessibility readings and the travel, but thank you so much. And thanks for everyone for participating today.